All right, here we go. It is the fall 2020. It is week 7. This is a big lecture today. This is gastrulation. Not so much that it's long or anything, but this is a very important subject, highly tested on boards. All boards, medical, chiropractic, physical therapy, acupuncture. Everybody has to know about gastrulation. So here we go. We're creating the germ layers today. I know you're so excited about that. All right, it's also, we have started week 3, so we're finally done with week 2, and we're starting week 3, or we're also starting the embryonic period. So, marks the beginning of the embryonic period, week 3. Some AKAs for that: organogenesis, morphogenesis, and this period will start at week 3 and end, it goes through week 8. And the star of the show in this very important week is a process called gastrulation. It's like Adele is the star of the show. Her voice is incredible, I think. So gastrulation. There's also, I should add, that there's a lot of other stuff going on simultaneously. This is a really big week in our little embryo. So it begins about, well, if it's week 3, it must be day 14, and that's when it usually starts. It doesn't perfectly start at that time. It may start day 16, 15, or 14, but on average this process starts on day 14. And yeah, we're at the beginning of week 3. The most important event of week 3, we already said, we are going to form the very important germ layers. These are all super stem cells. These 3 layers we're going to form, and they could create anything you want from these 3 layers. Now each 3 are destined to become certain tissue that we'll talk about as we go along through the remainder of this course. But if you could harvest any of these cells, it would be amazing what you could do with these things. All right, so the first step in gastrulation is the formation of something called the primitive streak. So that's where we're really going. All right, so the process, or the flat-out definition of gastrulation is the process of forming the primitive streak and then the trilaminar embryonic disc, during which you will create the 3 germ layers: the ectoderm, endoderm, and mesoderm. And here they are right here in our little a little cutaway view. And right now we don't have that, right? We have a bilaminar disc. And there's endoderm and ectoderm. Remember, we have a hypoblast, an epiblast, and hypoblast. So we're going to convert that epiblast into an ectoderm and convert the endoderm, or convert the hypoblast into the endoderm. And then we're going to create a brand new layer called the intraembryonic mesoderm. Everybody just calls it the mesoderm for short. All right, so a note here. So not all authors agree on when gastrulation stops. Some board books say that gastrulation is not over after you've formed the 3 germ layers. Our board books, our chiropractic board books more, he says, and other ones do too, that once the trilaminar disc is formed, that's it. But other board books go on to include the formation of the notochord as well as the neurotube. And after the neurotube is formed, gastrulation is over. So you have to check with your boards if this is actually going on YouTube. So if you're a medical student, you have to see who your board book is and see this is designed for chiropractors. Okay, so for chiropractors, gastrulation is the formation of the primitive streak and the trilaminar embryonic disc. So before we start, we got a little housekeeping to do. So we've been talking about our little bilaminar disc, our little human right now. We've been calling it a blastocyst. But once the primitive streak starts to form, a name change occurs, and now we're going to call it the gastrula. Gastrula. Okay, so just to recap, so we went from the morula to the blastocyst and now the gastrula. All right, and when in doubt, if you forget where you are, just call it an embryo. And that works pretty good. So goodbye blastocyst. All right, step 1, formation of the primitive streak. Let me get just a little water here. All right. So you can't do anything. You can't make the 3 germ layers without first making the primitive streak. What is the primitive streak? Well, it's a super important cluster of genes that happens in the caudal surface, the dorsal surface of the. We still have a bilaminar disc at this point. It doesn't form cranially. It's caudally, right in the midline. And these cells are magical. They have a lot of different genes turned on. And they don't really do anything per se. They don't grow into anything. But cells will migrate through these cells and come near them, touch them. And these primitive streak cells will release different gene products that soak into the cells as they pass by. And they manipulate their genes. They turn the genes on and tell them what to do and where to go and what to become. So these cells are special, especially something called the primitive node. It has even more genes turned on. In fact, the primitive streak has 3 parts. And what did I tell you about boards in particular? Boards only have 4, chiropractic boards anyway, they only have 4 answer choices. All multiple choice questions with 4 answer choices. And when you see something like this is perfect. It's so easy for a professor to make a question. Okay, so so primitive streak has 3 parts. Primitive node, sometimes called the primitive knot, or sometimes called Henson's node. It's kind of like the brain of the primitive streak. And then we have the primitive groove and the primitive pit, and we'll talk about that as we go through these things. Before we dig further, let's remember what we've built so far. So right now, here's our blastocyst. Hasn't formed a primitive streak yet. The primitive streak will be forming right back here in the caudal portion. That's what we're going to form. But right now, it's a bilaminar disc with a epiblast and hypoblast. Amniotic cavity, yolk sac here, really a secondary yolk sac. We formed these special AVE cells, which are going to come into play. We'll talk about what they do today. And then, then the rest is just hypoblast cells. All right, also remember that nodal is turned off. We made a big deal about these AVE cells and how they secrete. Their nodal genes get turned on and they secrete this gene product called nodal, and it soaks into all these cells up here, and when that gene product soaks into them, it makes sure that the primitive streak doesn't go there. So AVE cells went to great lengths to turn off nodal, so it shuts off, bottom line, it shuts off nodal in all these cells. And we made a big deal, and you'll see why in a little while. The AVL, to do this, how do they do that? What are the cells that are secreted? Did I say nodal? It doesn't secrete nodal, but it turns off nodal. We got the nodal turner offers. So there's 3 substances secreted by AVE cells that will turn off nodal in the cells above. And that's Cerebrus 1, Lefty 1, and Dickoff 1. We call it DDK 1. All right, those are the nodal turner offers. And yep, those are only in, let's see, that way the primitive streak grows back caudally. All right, induction of the blastocyst. Let's build the primitive streak. So the first thing that happens is the peripherally located, where's that peripherally located? Where's that overhead view? So the first thing that happens is, well, it's cut off, but these cells over in this region are going to start waking up and doing stuff. Okay, so peripherally located or laterally located caudal epiblast cells are invaded by nodal. So remember, these genes, these epiblast cells are still, nodal is turned on and they're secreting nodal. So nodal soaks in to those cells and it does some stuff. Okay, so nodal comes, let's see, where are we? Peripherally located, yep, so the laterally located epiblast cells are infiltrated with nodal. Nodal gene is turned on. And the nodal comes from the hypoblast cells below. So it kind of soaks into the epiblast cells. Specifically, nodal turns on some genes that induces or makes these epiblast cells migrate. They actually break their bonds and they migrate to the midline. Okay, and this only occurs in the caudal region. Remember, nodal's turned off in the cranial region. So, oh, here we go. I forgot I made this. So all the cells along the edge here, they wake up and they start migrating to the midline. This would be the midline of the bilaminar disc. And they start piling up right here into a pile of cells, and that's going to be our primitive streak. Right? So there we have our streak starting to pile up here. We also, we've just created an axis. Once they start piling up, that gives us, oops. We also know that this is the caudal end now because this doesn't happen cranially. So we know that this is the back end or the caudal end of the cell. That's where the primitive streak. So you could say we've just established the cranial caudal axis. All right, just another picture of it. Still have hypoblast and epiblast, but we got this weird thing going on here, the primitive streak being built. We got our caudal cranial axis going on. Remember, no nodal or BMP4 for that matter secreted up here, so that streak won't go in the wrong place. What really stimulates? We got to go a little deeper than this. What really stimulates? Because these are epithelial cells, right? They're bound together. So it's really a multiple. But let's talk about just the gene process first. It's a multi-step process. First, the nodal from below in the hypoblast cells. It turns on Wnt3 gene in these lateral caudal lateral epiblast cells. Then the Wnt3 wakes up the nodal gene in the same type of cell. So the nodal from below, these are epiblast cells. Here's hypoblast cells. Nodal gene is on in these hypoblasts. Nodal kind of floats up and it goes into the nucleus and it turns on the Wnt3 gene. Then the Wnt3 gene can turn on the nodal gene in these cells. Right? Then the nodal gene turns on other genes which we won't get into and it stimulates them to migrate to the center and pile up. We have a little more work to do, but we'll get to that in a second. Also, once the cells do start piling up, they don't pile straight up and up and up. They pile up a little bit, but then they start piling and moving forward. So the primitive streak elongates forward. Okay, it makes it to about halfway point of the embryonic disc and then it stops. Then the cells pile up at the end of it to form a primitive node. So if this is an overhead view of the bilaminar disc here, the primitive streak will grow this way, it'll grow and grow and grow and grow. And when it's about halfway toward the cranial end, this is cranial up here, it stops. But the cells keep piling up into a big bump. And that's the primitive node that's created. Primitive node is super, super important. There's a lot of smart cells up here. Some AKs for that primitive node, the primitive knot, Henson's node, or the organizer. You could also say that the primitive streak induced the formation of the primitive node, if you want, because the primitive streak grows first, and then cells move forward and pile up into this bump. And those cells are magical up here. They have all sorts of special genes turned on. We'll continue to talk about that. But so there's a real better than I could draw. But there's what we've created so far. Primitive node is like an aircraft control tower. It's all sorts of genes turned on. When cells pass through the region, just like any part of the primitive streak, when cells pass through it, they're kind of programmed to do things and go to certain places and become certain tissue. But this region's especially important. Right? What are a couple special genes that are turned on here in the primitive node? Well, you got to know these guys. The goosecoid, cordon, and FOXA2 genes are particularly important. They're only initially on. We're going to turn them on in other places too in the primitive streak, but right now they immediately get turned on. Goosecoid is really important for neural induction, forming the neural plate. So we'll talk about neurulation. We've got to have the goosecoid gene working for that. It also activates cordon and another one called noggin. And cordon and noggin are important for sidedness. For example, they secrete gene products which tell the heart that it's supposed to be a little bit more on the left side and not the right side, and things like that. Cordon, which we just said is turned on by goosecoid, that's also important for formation of the neural plate and neural tube. So neurulation, goosecoid and cordon are very important for neurulation. FOXA2 is really important for maturing the primitive node itself. So that's important. And it's also vital for formation of structures that are in front of the node. Prechordal plate we'll talk about toward the end of this lecture. That's all about FOXA2. And even more importantly, this is the first gene that's super important for forming the nodal cord. And that's what we'll talk about in the next lecture. These migrating epiblast cells continue to come to the midline, so they're piling up. Even though the primitive streak is formed, epiblasts keep piling in, and as we said, the cells will migrate. Let me back up. Once the primitive streak and primitive node are formed, these epiblast cells just don't stop. They keep coming and migrating to the streak, but they're not going to create a new structure. They're not going to make it bigger. Instead, this kind of the second wave, if you will, they're going to pass underneath it and through the streak. Right? But the original streak cells aren't going to move. But as they pass by or through this primitive streak, in fact, these cells passing through start to create a groove called the primitive groove. In the primitive pit, these cells keep going down through the streak and creating these structures. But they're going to get all sorts of genes turned on when they do that. All right, they'll also, let's see, what else? Get back on my slides here. They'll migrate through and under the streak to create new structures. Great. They'll also migrate down and through the primitive node. And then another batch of them will actually move forward in the midline. There's 3 waves of cells that will actually go forward. So we'll talk about those. And here's a good little picture here of what's going on. So the streak and the node are already built, but we still have cells, and they're modified epiblast cells. We have to back up a minute and explain how they get loose. But they migrate right through the streak. They're told what to do, and then they take off in all directions. These green ones here are going straight forward. This first wave is going to create something called let's see, what do those first ones do? The prechordal plate is what this first wave is going to become in a second. These pink ones are going to spread all over. They're going to come right at you out of the plane of the page and deep into the plane of the page to start making a mesoderm tissue, as we'll see. Okay, so more and more epiblasts keep migrating down through the primitive streak, and they start leaving a dent in the streak, and that's the primitive groove. They start passing through the primitive pit as well, and that leaves a hole. And that's called, or they start passing, so many start going through the primitive node that it gets a hole in it, and that's called the primitive pit. Primitive pit is actually continuous with the primitive groove, and that's about all we'll say about that. You can see it how all these migrating epiblast cells have literally put a dent in this thing, and a dent here as well. So primitive pit is inside the primitive node. Primitive groove is a groove from cells diving down through the cells of the primitive streak. Electron microscopic view here. You can see the primitive groove quite nicely, and there's the primitive pit. All right, so primitive streak, with a primitive streak going on, we already said really we know the cranial and caudal axis, but we also know where dorsal is. We know where ventral is now because the primitive streak grows on the dorsum. It grows on epiblast or epiblast cells, so we know that's going to become the back. We know the ventral. We know left and right, so we know quite a bit about the direction of our little creature here. All right, so left and right, and we know lateral, we know dorsal. The caudal would be underneath, deep into the plane of the page. I think you get that. All right, now the primitive streak is formed. We're ready to start making the germ layers. No, we're not, because remember these, we need to back up and talk about some more stuff. Remember these primitive, these epiblast cells, they're epithelial cells. And epithelial cells, Dr. Doe taught you, there's cams between them. There's cell adhesion molecules that hold them together. So what's the deal with that? They can't migrate anywhere, so we have some more work to do. So let's back up. Epiblast cells are columnar. They're columnar epithelial, simple columnar cells, and therefore they have very strong junctions between them called cell adhesion molecules, or you could call them junctional complexes. J.C. Ross, the histology book, uses that term. Others use CAMs. But we need to break those things because they're not going anywhere. Then, after we break them, we need to, because we're going to make a new layer between epiblast and hypoblast, we need to make that, put some oil in there and make it slippery. We're not putting oil in there, but we're going to secrete some substances that are very slippery. Here's Dr. Doe's slide. You're like, oh no, not this again. Yeah, even when in fifth quarter we'll keep talking about these cams. Can't get away from them. Tight junctions. Remember the tight junctions up here, they're no good at holding cells together, but they're great at stopping things from trying to get between cells, like acid in the esophagus. So they're important. But the things we need to break are these zonula adherens, these guys right here, and desmosomes, aka macula adherens. The cells can't go anywhere until we break those. So that's exactly what does that. Now, warning, Mr. Spock is like, what? The authors are all over the place on this. So there's just not enough human embryos to get the full story. But so this is based on chiropractic board books, this story. So the breaker of chains, right? This Game of Thrones theme when I created this PowerPoint series. So I threw her in there. She's the breaker of chains. And so we need to break these complexes up. So epiblasts are locked together by two types. We said that already. They have to free themselves. We already said the main types are zonal adherens, macula adherens. Come to find out, these are both from these kind of rivet type things are both made from the cadherin family. The ecadherens is what makes up zonal adherens. The desmoclines and desmocollins, which hold desmosomes, or which desmosomes are made of, are really also from the cadherin family. So if we could get one biochemical that breaks these or shuts down the production of these, that would be great. And that's exactly what happens. And enter our friend, the snail. Don't forget the snail, very important. You wouldn't be alive without the snail. So breaking cadherins is accomplished by the expression of a transcription factor called snail. And so snail has to be turned on in these epiblast cells before they do anything. And somehow, we're not exactly sure how snail gene turns on. The thought is it's nodal that somehow does it. We're not 100% sure, but snail is on. And with snail on, it turns off the genes that create the cadherins. So those cadherins don't last forever, and they have to be replaced. And if you turn off their gene, pretty soon they're free to migrate. And that's exactly what happens. Right? And just these are the adherent junctions and the desmosomes, and you could see the desmocollins here, desmoglines here, and you talk about that more in histology. There are some talk about that about pemphigus vulgaris is a wicked, horrible, blistering disease of the skin where you get an autoimmune attack against these, but we'll talk about that in 7th quarter in dermatology. Alright, so that's the story with that. Alright, so now we need to make it slippery, the space in between the ectoderm and endoderm, or that's going to be the ectoderm and endoderm. Right now it's the epiblast and hypoblast. We need to make that slippery. And so these epiblast cells, they also turn on the genes that make hyaluronic acid, which is a glycosaminoglycan, and they make fibronectin, which is a glycoprotein. And especially hyaluronic acid. And did we talk about that? Yeah, we talked about that in spinal anatomy already, how slippery that stuff is. Gags are really slippery. And so yeah, so that stuff goes down and it coats the migrating epiblast cells, get this slippery stuff all over them. And then it slips down between the epiblast and hypoblast and makes that area really slippery so cells can squeeze in there. Now we're ready to migrate. So let's get migrating. And another thing, so these are still simple columnar cells. So they can start working their way toward this primitive streak. But in order to dive down underneath the streak, they have to, well, first of all, they have to have some ability to move. So we have to grow some arms on them so they can crawl. So they're going to kind of metamorphosize as they start to pass through the streak. So in order for epiblasts to migrate, they have to morph, or metamorphosis has to occur. And they first morph into something called a bottle cell. Some people call them flask cells, but the official word is bottle cell. And they are pre-mesenchymal cells. Mesenchymal cells are like stem cells, not quite as powerful. They have a more destined role, so they can't turn into any, you can't harvest them and make any tissue out of them, but they're very stem cell-like. But the bottle cells are not quite there yet. Here's a good picture. So epiblast cells are going, and I mean, this is primitive streak cells. They don't move. These would be going underneath the primitive streak cells here. But as they pass underneath, they morph into this weird bottle cell, which immediately morphs into a full-blown mesenchymal cell. Mesenchymal cell has pseudopodia, little arms that move, and this can crawl its way between the, see, look at all these, they didn't draw them all correctly, but these are all mesenchymal cells that are crawling their way between the ectoderm and endoderm and forming a new layer of cells, and that's the mesoderm here. Okay, so that's really important. So epithelial-to-mesenchymal transformation. So the bottle cells that are now passing through and beneath the primitive streak and epiblast, they morph into mesenchymal cells, and they grow pseudopodia. So now they can crawl. We talked about that already. But the point is, once they're converted into mesenchymal cells with little arms, a process has finished. So we took epithelial cells and we converted them into mesenchymal cells. So that's called the epithelial-to-mesenchymal transformation. That has occurred. And mesoderm, that's it. It goes through this process and it doesn't turn into epiderm again, or epithelial cells again. It's not true with the endoderm, as we'll see. All right, when we talk about neural crest cells, they also go through a process of epithelial to mesenchymal transformation. So you'll hear that again. So now finally we're ready to go. Who tells the mesenchymal cells where to go? Well, there's two main genes turned on in primitive streak cells that help direct them. And these genes basically tell the epiblast, bottle cells, and mesenchymal cells where to go. So here's the little scary man, so hang on, bear with me. So we have the brachyury gene. Sometimes it's called the T gene, which is the first original member of the T-box family of transcription factors. So what does it do, this T gene or the brachyury gene? It activates something we already know, FOXA2, right? We said FOXA2 is for nodal chordal formation. And goosecoid, that was for neurulation. So it turns those genes on in the primitive streak cells. So now FOXA2 and goosecoid are not only in the primitive node, they're in the primitive streak as well. Great. Let's see. They also protect bottle cells from passing through the primitive streak. I guess it can be kind of a rough trip, so it makes them a little tough. And then we have nodal. That, of course, nodal's got to be in there. So nodal tells the migrating cells where to go based on how much nodal they're exposed to. So when a cell passes through, or when the cells passing through that receive high doses of nodal, they have enough power to go all the way down to the hypoblast and kick those cells out and become endoderm. Cells exposed to low levels of nodal, they don't have as much power to go all the way to the endoderm. They have enough power to get underneath the epiblast cells, and they can become they'll become the mesoderm. Did I say that right? Cells exposed to a high level of endoderm have enough power to kick the hypoblast cells out and make a new layer called endoderm. The cells passing through the streak that get exposed to low levels of nodal, they don't have the power to go all the way down to the epiblast or hypoblast layer. They just have the power to go underneath the epiblast layer, that top layer, and squeeze in between the epiblast and hypoblast, and they become the mesoderm cells. All right. Yep, we already said they're ready to go. Everything's slippery. So now the process, the migration is starting, and that of course has a name. It's not called the Great Migration. That's me talking. Officially, this is called ingression, and you've got to know that word. Ingression is the beginning of the germ cell formation, We're making the trilaminar disc with this process. All right, prepared epiblast cells can now migrate downward through all regions of the primitive streak and primitive node in a process called ingression. Langman, which is a chiropractic board book, unfortunately calls it something else. It calls it invagination. Moore, here's the other chiropractic board book, calls it ingression and doesn't call it invagination. I know, drives you crazy when these authors can't get on the same damn page. It drives me crazy too. Makes it a lot harder to build PowerPoint presentations, but what am I going to do? So unfortunately, those are very strong AKs of AKAs. All right, so there's the story again. We kind of back to that sneak ahead. So we got all these bottle cells morphing into mesenchymal cells, being told what to do. Foxa and Goosecoid and the gang, nodal, is telling them where to go and what to become, and they're literally pushing the hypoblast layer out, and they're going to start filling up, creating a new layer. And remember, this is a two-dimensional picture, so these guys are going deep into the plane of the page. They're coming out of the plane of the page right at us. Some are heading straight up here. All right, so this is controversial. Some authors say the endoderm forms first. Some say the mesoderm forms first. Carlson says they form at the same time, which I believe that. I don't think we really know 100% sure which way it is. But for you chiropractors out there, mesoderm is going to form first. And we can see these cells are moving, going under the primitive streak cells, being programmed, and off they go. And if they got a high dose of nodal, they'll go all the way down here and start kicking these hypoblast cells out. If they got a low dose of nodal, they don't have enough power to go way down there. They start filling in the middle here. Those will become mesoderm cells. So second will be the endoderm, as what we're, and I already just said this. So if they got a high enough dose of nodal, do I have that written? Yeah. Cells are exposed to high levels of nodal, so they go through the mesoderm layer, and they start kicking out these green cells, and those were our friends, the hypoblast cells. Remember, we said way back when we were talking about forming the extraembryonic mesoderm that possibly cells from the primitive streak formation were going to be involved in that. So that might kind of finish off the extraembryonic mesoderm formation. But these new pink cells are our new cells, our new mesenchymal cells. And guess what's going to happen to them? They're not going to stay mesenchymal. They're the ones that form the mesoderm. They're not going to turn back into epithelial cells, but these guys are actually going to morph back into epithelial cells, as we'll see here in a second. So endodermal cells convert back to epithelial cells. So yeah, so those, just what I said, the mesenchymal cells, they go down into the endoderm, they're converted back into endothelial cells. So cells of the new endoderm, which we just created, they actually went through a process called epithelial-to-mesenchymal to epithelial conversion. All right, so epithelial, when they were epiblast cells, they morphed into mesoderm cells for their journey. They kicked those hypoblast cells out of there, morphed into new endoderm cells, and now they're converting back into epithelial cells again. Got it? All right. So there's what we have so far. We have this mesoderm. These are still mesenchymal cells. They haven't changed. Then we have these new endoderm cells, which have morphed back into epithelial cells. And now the last layer is the ectoderm. So we don't know a lot about how this happens, because these were the cells that created everything, right? These ectoderm cells, or these epiblast cells. But while this process is going on, a metamorphosis is also occurring in some of the epiblast cells that stay behind. And they morph into a new cell type called ectoderm. All right, let's see. And so once they morph into this new cell type, we have the three germ layers. We have the three new germ layers: ectoderm, mesoderm, endoderm. What does ectoderm become? A whole bunch of things, and we'll talk a lot about what all this stuff, and that's for boards and medical boards. I think everybody, one of the keys of embryology is to learn what this stuff is going to become. We're going to fold this into a tube later on, but you got to know what this stuff becomes. But I mean, epidermis is probably the best answer. Ectoderm becomes epidermis. Not all regions of the body, but all right. This is my thing about mesoderm or ectoderm. What really comes first? I mean, chiropractic, the board books, Moore says that the mesoderm is created first. Larsen, which is more European book, he says the endoderm's created first. And Carlson says they probably happen together, which makes a lot more sense. But we're going with, for chiropractors, we're going with intraembryonic mesoderm is what's created first. Okay, if you're a European medical student or chiropractor, I don't know what they use. Larson over there is a big book, really well written book. I really kind of my go-to book. Maybe not quite as deep as Carlson is, but very good storyteller. Here's how he describes it. He believes that the endoderm forms first. Once that's filled in, then the mesoderm comes down second. But that's not what we're going with for us USA chiropractors and Canadian chiropractors. We're going with mesoderm goes first. How about some fun facts? We got to do fun facts. It should be noted the ectoderm forms in the cranial region first. So the epiblast cells start morphing into ectoderm away from the primitive streak. See why that's so weird? Because remember, the primitive streak is in the caudal, and the ectoderm forms first cranial. So that's kind of a mystery why that happens. And then it kind of sweeps backwards. So at least with regard to formation of the ectoderm morphing into, or epiblast morphing into ectoderm, that process starts cranial and sweeps backwards. Another very important thing is to note that these epiblast cells are super magical. They gave rise to all three germ layers. The epiblast cells, if you're a mad scientist, these are the ones you want to get. If you want, because you could grow any body tissue with these epiblast cells. They are magic. All right, let's talk about some more work, because we really have we finished gastrulation? There's some more stuff that forms during the process of gastrulation, so we need to back up again. We have to talk about the prechordal plate. This is very, Larson says, is one of the most misunderstood structures in human embryology. Prechordal plate, head organizer, Carlson calls it. A prochordal plate, Singh, that's an India author. And we still don't understand the deal with this thing. So authors are all over the place. The monkey's hairs, I guess that's not a monkey. Is that a ape or a chimpanzee? I don't know what that is. Orangutan, whatever it is, its hair is gone mad. So even our chiropractic board books, Moore, which is one board book, says the prechordal plate is a thickening of the hypoblast. I don't think that's true, Moore. So he needs to rethink that. Langman says it's a collection of mesoderm cells. Carlson tends to agree with that, so that's what we're going. So the prechordal plate is made from mesoderm cells. It is formed between the epiblast and hypoblast. So the first of three, remember I said there's going to be three waves that migrate forward of epiblast cells? And now we're backing up. We haven't made the three germ layers. We're still making them now. Some of the cells go through the primitive node and they go straight forward in the midline. We're going to see three waves of those. And this first wave, they start to pile up to form a plate, and that's going to be called the prechordal plate. And it migrates between the epiblast and hypoblast cells. It's the first migrating wave, passes through the primitive node, and the migration stops toward the cranial end, doesn't go all the way cranial, but they pile up and they form a plate, and that's going to be called the prechordal plate, or head organizers. So let's see what's happening here in a drawing. So here's primitive streak. We got the mesoderm starting to form over here. Some of these cells go right through the primitive, down the primitive pit, through the primitive node, and they go straight forward. And we can see these little pink ones have been piling up, and they've been pushing apart the epiblast and hypoblast. There's the AVE cells we've already created. They kind of overlap that a bit. That's our prechordal plate. All right, there's a view from overhead, kind of a transparent ectoderm, or I guess epiblast. We haven't made the ectoderm yet, but you can see this. Here's what's going on underneath the ectoderm. Mesoderm is being made. Its cells are sweeping all over this. And some of these are diving down into the plane of the page to make endoderm. But some of these populations are going straight forward and piling up in this weird plate. That's the prechordal plate. Why do we care about this prechordal plate? Well, it forms your brain. It's one of the first signaling centers that tells more epiblast cells and other types of cells we'll talk about later to go up and form your noodle. So the forebrain is the first part of the brain formed, and there's an important gene that has to be turned on here. This gene is not turned on anywhere else yet, and that's our friend the sonic hedgehog gene. Okay, so this new mesenchymal cells will migrate through the prechordal plate. As soon as they pass through, and this is after the prechordal plate is formed, the sonic hedgehog gene products will tell them to go form the forebrain. Okay, so a little bit more housekeeping to do before we can call it. Gastrulation is still not complete. There's a caudal membrane, or a cloacal membrane. How it forms is all over the place, and we just don't know for sure. But we know about the same time the prechordal plate pops up cranially, another circular plate forms caudally back in the midline. And it looks like the epiblast and hypoblast cells pinch together and just squirt the mesoderm cells that are forming away from it. So this cloacal membrane is only 2 layers thick, and that's a testable question, because the rest of the embryonic disc, the little human, is 3 layers, but not here. And we're going to see another twin structure to this in a minute. So cells packed together, whatever happens there, there's no mesoderm there at all. And so the cloacal membrane is a bilaminar plate, even after the trilaminar disc is built. Okay. And there's what it looks like, kind of an overhead view of our bilaminar disc. This author was nice enough to remind us that this view, we're looking through the amniotic cavity. Right? Underneath that would be the secondary yolk sac. Let's not forget we have a ball here. This is all going on kind of inside the ball. But there's the cloacal membrane. What's the big deal about this? This is going to become the anus, the end of the GI tract, as well as the urethra in male and female, and the vagina even in females. Right? Those structures, we won't get to those in this course. That's way up in week 7. And yeah, you could say the cloacal membrane is the terminal end of the gut tube. You sure could. And now a twin structure called the oropharyngeal membrane. You need to talk about that. So about the same time the cloacal membrane forms, another circular structure forms just same way. the ectoderm and endoderm seem to pinch together and kind of squirt the mesoderm out. So this is another bilaminar structure in a trilaminar disc. And it forms just in front of or cranial to the prechordal plate that we just made. So prechordal plate we said was mesoderm, so that would be underneath this view. But here is the pinching of these two layers together. That's the oropharyngeal membrane. Guess what that, if this is going to become the anus or the end of the GI tract, we'll take a wild guess what that's going to become. Oral. That's going to become the mouth. Okay, so yep, everything I said. Bilaminar portion, the mesoderm is squirted out, and this forms a little earlier, about the week 4 or so, and that's, you could say, the start of the foregut. So now we have all these structures done, right? We have a primitive streak, and they all get done about the same time. We have an oropharyngeal membrane, we have a prechordal plate, and we have our cloacal membrane. Gastrulation is complete according to most authors. And anything else I want to say? Yeah. So technically gastrulation is complete. Why? Because the primitive streak is formed and all those associated structures are formed. And also, remember though, check with your board if your other medical students or whatever watching this. Check, because some authors say that we need to go one step further before gastrulation is completed, but not our chiropractic board author. So we are done with gastrulation. We'll talk about formation of the notochord and nerulation next. Hope you enjoyed it. See y'all later.